As you know, I'm a cardiologist in an intensive care unit, and I'm seeing all these problems in medicine, and I hope that nanomedicine will give us a big boost, and I'm convinced so, but we have big gaps to fill. So we all agree that nanomedicine in its young life has already achieved some very nice results. For example, in targeted therapy, we see pharmacological innovations like today and yesterday, but what we also see is that in preclinical trials with animals, we always see these nice slides showing cures of cancer in animals. We also see very few clinical trials where nanomedicines have prolonged survival. But do we see cures with nanomedicines in humans? I think this topic of translating cure of disease from animal experiment to humans, this is a key goal we should achieve, and which is needed to show that nanomedicine is not just a little better than conventional anti-cancer drugs, but that it's really a new class of a new concept, a new notion of therapy. We have also to mention that certain clinical trials have been stopped because of side effects. You won't see those trials in any publication because of a publication bias. It's always more difficult to publish negative results, but also because of commercial concerns about specific companies. This problem of publication bias in clinical trials hinders our understanding and our advancement to figure out where the real problems are. And I strongly would encourage you, even if I know that it will not help a company, that we should really discuss also about the problems and not about only the successes of such nanomedicines. We have also seen that nanomedicine is applied a lot in cancer. This is because cancer is a very bad disease, because people die of cancer, but there are many more diseases than cancer. And what we also would like to see is breakthroughs in other diseases. In the other diseases than cancer, the requirements for drugs are different, and they may be more complicated. If you have a 5% side effect rate in a drug, it may be well suited for cancer therapy, but it may not be suited at all for hypertensive, antihypertensive therapies, etc. Now, the big challenge for medicine is we have not mastered any of the big killers in medicine, neither infections, cancer, arteriosclerosis, diabetes, brain diseases. So there is a lot to do. And I hope that in some of these diseases we can really make a difference. But another big problem is the cost of healthcare. If we look at the problem of medicine and of nanomedicine development from a company point of view, the question is always how can we increase our income, the money we make with a new drug. But on the side of the patient and society, there is a real problem today with financing of healthcare. And the question is, we know that the costs will rise more and more because of the aging of the population, because ineffective therapies are still widely applied, and because chronic disability increases if we cure certain diseases at the stage where they keep the patient alive, but do not remove his disability. That means one of the big challenges for nanomedicine is not only how to produce more money for a company, but how can we make medicine more effective and less expensive. This is particularly important for certain areas of the world where the amount of money that can be spent for health is very low. And I remind you of the current efforts to eradicate malaria. The eradication of malaria slowly moves forward, but it cannot be achieved if we don't achieve major progresses in diagnostics and in therapy, in particular in the financial aspects of diagnosis and therapy. So the challenges of a sustainable medicine is there in developed countries and even more in developing countries. For example, one challenge is rapid testing for malaria. Current testing is often done by lateral flow assays, which cost about 50 to 60 cents per test, which is more than you spend for the treatment of a child with malaria. Now, to eradicate disease which becomes less prevalent in the course of eradication of malaria, you need to do more tests on patients who will end up with a negative test. That means testing per positive case becomes more important. So what the malaria community would need is a test that is 10 times less expensive than today, 5 cents per test, but is better than today's test. So high quality, high sensitivity, stable at 45 degrees storage over a year, built-in positive control and quality controls. Can we achieve that with nanomedicine? Maybe the pieces are there, but I don't see the efforts in really going that way straightforward, which would make a big difference for mankind. The other challenge I see from a clinical perspective is the challenge of fundamental understanding what we are doing. We see a lot of advances at the physics side and at the basic pharmacologic side of materials. But what is still lacking is a more generic understanding of physiology. What does a human body do with nanomaterials? How do the differences in nanomaterials influence physiology? Most nanomaterials are either short circulating, they are rapidly eliminated by the liver and the kidney and the spleen, sometimes are so-called long circulating, but that also means that they are eliminated after 48 hours by the liver, and very few have a longer half-time. We have the problem of stealth versus targeting. If we produce nanoparticles which have excellent stealth properties, we have a good chance that it's also difficult to have a ligand on it which then really binds to the tumor. And then an important point below, complement activation related pseudoallergic reactions. We'll hear more of that later. Now, targeting and stealth are interdependent and sometimes antagonistic. We need long circulation time for optimal targeting. At the same time, protein repellent stealth properties may hinder ligand-receptor interaction. And one thing we often forget is that the long circulation time facilitates cargo unloading before arrival at the target. Now, the unloading of hydrophobic substances from their carriers is a physiologic process. Most hydrophobic molecules in the body are carried by some carrier like albumin, etc. And the physiology means that they always exchange their payload. That means how can we guarantee that a long circulating carrier, which is excellent for targeting, does not lose his payload until it arrives at the target, but then at the target can still unload and has not too stable attachment between drug and carrier to remain fixed on this position. That means we need a systematic predictive nanophysiology, pharmacokinetics, nanotoxicity in humans, and not only in mice. But my suspicion is a little bit that the results we are showing here, that if you inject a fluorescent labeled nanocarrier, which strongly goes to liver, lung, spleen, kidney, that the very same results are true for many nanoparticles in preclinical phases when we apply it to humans. This is the story of liposomal therapies. So we have had a long, big expectation of liposomes coming to the clinic, but the reason why so few liposomes have become blockbuster therapies in medicine are twofold. One, because liposomes, to the most part, have not mastered this challenge of premature elimination. And second, because liposomes still come with side effects like these pseudoallergic reactions. Composite therapies behave different than single molecules. This is, for example, shown in this pharmacokinetics data we have achieved with polymeric carriers. And the reason behind that is you have an object which is composed of multiple blocks, and there is an equilibrium because the nano object composed of blocks and unimers which also can circulate in an individual fashion in an equilibrium relation. In our case, for example, this leads to a two-phase renal elimination. In principle, you have a polymeric assembly of a nanomaterial, which means that the individual unimers can be filtered directly through the kidney. On the other side, you have the assembly as such, which has a kinetics of disassembly, which leads to unimers which can then later be eliminated by the kidney. So what we are waiting for is a predictive pharmacokinetic modeling of nanomaterials in humans. One example of this is our Basel virtual patient, which is a simulation environment of multiple organ systems in which we try to use experimental data for parameter estimation of pharmacokinetics model in the hope that we achieve a point where we can predict the behavior of future drugs for rational design of new strategies and materials.